Hello everyone, welcome to Practically's NEET Bio class, and today we're going to discuss about cell cycle and mitosis. So, new day and new topic. Today is basically we'll be discussing about different kind, like what is cell cycle, the basic idea. Okay, cell cycle and mitosis comes under cell division. Okay, how a cell divides. We know that a cell divides. Okay, whenever the cell has reached its saturation, it has to divide, it has to become into a second cell, it has to give birth to a daughter cell. So today's basically we'll be discussing about the cell cycle, how the division happens, and we'll be discussing about mitosis. Now in eukaryotes there are two kinds of division, which the mitotic phase, whatever it happens, there are two kinds, there's mitosis and meiosis. Okay, so today we'll be discussing about mitosis only. So let's get started, put our thinking caps on and start. Before that, we'll discuss a little bit about Practically. We are from Practically and we have a very interesting app which you should definitely download. It has everything you need, all the scientific courses, maths, physics, chemistry, bio, everything which runs for 6 to 12. There's a lot of simulations. You can ask there all your doubts there directly, and I'm sure you'll get it answered. Okay. Apart from that, you can also follow us on our socials on Facebook and on Instagram. And do not forget to like, share, and subscribe to our channel. There's also a new channel which is solely dedicated to 11th and 12th NEET. Okay, do check that out as well. Now we were discussing about what was happening. Okay, let me see. Okay, so we have some people around here. Hello everyone. Hi Shangship. If you have any doubt, please just leave it in the chat box. Now, so why is this cell division happening? Before, just now, as I said when I started the class, that when the cell reaches a saturation, okay, that time the cell has to divide. Okay, because it has reached a saturation. After that, it is full of, like, it is full of nutrients, it's full of DNA, RNA, it's full of biomolecules. Okay, now it has to divide to give birth to a daughter cell. Okay, so that is why cell division happens. Now the thing is, we know that one cell is coming from a different cell. Okay, coming from the mother cell. A cell always has a mother cell. It does not just grow out of it on its own. So the thing is, it just comes out of a mother cell. So why? Because a cell born after a division proceeds to grow, okay, by macromolecular synthesis, divides, and after reaching a specific determined division, its size, it divides. Okay, now again it will grow again. Okay, it will grow and it will have a number of proteins, it will have its genetic material, everything it will divide, and everything again it will give rise to another cell. So it is like a cycle. Basically, it is like a cycle. It is a cyclic process. Okay, so what happens? There is one cell, it's growing. When it has grown properly, it has grown, it has all its whatever it's needed, it divides into two. Now these two again will grow, these two daughter cells will again grow properly, again they will also divide. So like this, a cell keeps dividing. Okay, that is why we are growing, right? When we were small, we had lesser number of cells, we grew. Okay, every year we used to measure our height, and we saw that we were growing. As our age grew, we started growing. Okay, we grew. When we started growing, first we were starting to grow in a height wise. Okay, then we started growing in a width wise. Okay, so that is how, in any way, in any scenario, we keep growing because our cells keep growing. Okay, so this is a cycle. That is why the cell division happens. Now, what is the cell division? Okay, so it's the process by which cell divides to form new cells, as I said, from mother cell to daughter cell. Okay, so there are basically three types of cell division. Okay, one is basically for prokaryotes. Now the thing with prokaryote is it is a single cell. It does not have a number of, it is not multicellular. It does not have a number of cells in its body. No, it's just one cell. Now that one cell, okay, when it's dividing, that themselves for them is when they're dividing, that themselves for is reproduction. Okay, so for them prokaryotes, okay, they reproduce via cell division. Okay, and that way it is known as binary fission, where the mother bacterial cell will give birth to two daughter bacteria. Okay, now for eukaryotes it's a bit different because we are mostly multicellular. So we are multicellular. It's not that only one cell will give birth to two daughter cells and done. No. So it is kind of a complicated process. So eukaryotes have mitosis, where the cell or growth of organism, replacement or repair of damaged cells and all, mitosis happens in these places. Okay, and meiosis is basically for the formation of sex cells or gametes. Okay, so this is basically for growth, repair, damage, etc. This is for reproduction. Okay, so today we'll be discussing mostly about mitosis, and in the next class we'll be discussing about meiosis. So mitosis, as I said, is a bit less complicated than meiosis. Here there is a mother cell, okay, and it divides, it changes its structure, internal structure, and it then divides into two daughter cells. Here, in mitosis, there are two daughter cells forming. In meiosis, it's a bit complicated and there are four daughter cells. Okay, so there's a division which we'll discuss in the next class. Now, so first a little bit about the prokaryotic cell. Hi Neha, good evening. Thank you for joining again. Okay, so first we'll discuss about the prokaryotic cell division. This is basically for bacteria. Okay, this is for no one. This is just for bacteria. As I said, bacteria is just one cell. Okay, it's not that it has a huge body or anything. It's just one cell. So if it's dividing, for it dividing means it's reproducing. Okay, so there are three major steps here. First is the DNA replication. Whatever, it's not that it has a very high-pi genetic material or anything. It has DNA. That is the genetic material that is just floating in the cell. Okay, so DNA first has, DNA is copied, resulting into two identical chromosomes. Okay, see here, there is a mother and there'll be some extra daughter ones also. Okay, then the chromosomes separate and move towards the end of the poles. Now they'll separate. Okay, one has come here, the other one has come here. And then the cytoplasm, okay, the external material, whatever external material is there, they start dividing. Okay, finally, they divide and form into two different cells. So very easy. There is the mother, which has a little bit of genetic material. Now this genetic material doubles. Okay, then it starts dividing. And finally, after the whole cleavage thing is done, there are two cells. One is the mother, one is the daughter. Okay, so now the best thing here, the thing which you have to remember is each new daughter cell is genetically identical to the parent cell. It is a ditto copy. Okay, the thing is the bacterial mother cell and the daughter cell is a ditto copy. Okay, there is no difference. It looked just the same. Because it is just the breaking up of a cell, no? It is just increase of the genetic material and breaking up of the cell. So how will it look different? So it is a copy. Okay, so that's it. That's it for the prokaryotic cell division. Eukaryotic cell division, as I said, is a bit more complicated because eukaryotes are a different kind of a structure. There are more organelles. The genetic material is covered by the nuclear envelope, and they have multiple cells. A body has multiple cells. We are eukaryotic. We have multiple cells. So the process is a bit different. Okay. So here what happens? Cell division that results in two daughter cells, each having same number and kind of chromosomes as the parent. The thing is, here it's not identical. Okay, they are not properly identical. They have same kind of structure, same kind of things, like same kind of genetical structure and all, but it's not identical in nature. It's genetically identical. Okay, so when there is mitosis, as I said, mitosis happens for growth, for regeneration, for damaged cell and all. When there is mitosis, okay, there are basically two steps. There is mitosis, which has 4 steps: prophase, metaphase, anaphase, telophase, and there is cytokinesis, where the cytoplasm divides. Okay, and the daughter cell is genetically identical, not fully identical, it is genetically identical. Okay, and meiosis, we'll be discussing about it in the next class. Now, before we jump right into the whole cell cycle and division and mitosis and everything, there are some things, okay, some terms which you have to definitely remember, which will help you in the next chapters as well. Okay, so the first thing is chromatin. Okay, now in the last classes, when I was discussing about nucleic acids, I told you that the DNA, or basically the DNA, which is a genetic material which is passed on from mother to daughter, for parents to children and all, okay, we have a huge amount of DNA in our body. Now everything is actually wound up very tightly, okay, because it's a huge amount and it gets wound up very tightly, okay, with the help of something called histone proteins. Okay, so what happens? What is a chromatin? It is a complex of nucleic acids and proteins in the cell nucleus that stains readily with basic dyes and condenses to form chromosomes. So what happens when you take that DNA? Okay, see here in this picture, you can see you have the DNA, and these round things are histone proteins. Okay, so when you take that DNA and you wind it up around the histone proteins, you form a fiber-like structure. Okay, so that fiber-like structure is the chromatin. Okay, so this is just DNA plus histone proteins. Okay, now what is chromatid? So either of the two daughter strands of a replicated chromosome that are joined by a single centromere. See here. Okay, so when this chromatin comes together and forms the structure of chromosome, there are two basically. Okay, this is one and this is another. So each of them, if you divide it, each of them are known as chromatids. And chromosome is the whole structure. Okay, the whole chromatin fiber which holds up and which makes up the chromatids, and it is joined with the centromere and everything. Okay, the whole structure is known as a chromosome. Okay, so it is what? It's a thread-like gene carrying bodies. Why gene? Because it has DNA. Okay, it has the DNA. DNA is carrying the information, the genetic information and everything. Okay. And their chromosomes are composed of DNA and protein, DNA and histone protein. Okay. Now, the thing is, they are visible only under magnification during certain stage of the cell division. Only during certain stage. As I said, the division has stages. Okay, during certain stages only, you can see the chromosomes properly. Okay. Now, what else? What else should you know? Okay, what else word should you know? Next is the spindle fiber. Okay. Now, when the cell, we'll discuss about it just after this. When the cell division is happening, when the mitosis phase is happening, okay, these chromosomes are held with some kind of filaments in the cell. The chromosome are held with some kind of filaments or fibers. They are holding on to somehow, because these things are moving, the chromosomes, the chromatids, they're moving. Okay, so somehow they have to work on a, what to say, on a road, right? They have to work on a road, they have to walk on a road. That kind of road is provided by the spindle fiber. Okay, so the network of filaments that extend inward for the poles of dividing cell, forming a spindle shape. This is a spindle shape like this. Okay, so this is kind of a road on which the chromosomes are traveling. Okay, centromere is whatever the chromosomes they were holding on to it, the most condensed and the constricted region of chromosome to which the spindle fiber is attached. See, now as I said, the chromosomes have chromatids, they are held together by something, no? This thing attaches to the spindle fiber. So this area is known as centromere. Okay, so this thing which is holding the chromatids and the spindle fiber is the centromere. The last thing is equatorial plane. Okay, this is just something which is seen in the metaphase. Okay, we'll discuss about it after this. Okay, where the chromatids, okay, they are sitting on a single plane. They sit on a single plane. That is known as the equatorial plane. We'll discuss about it. Okay, let's see who is there. Okay, hi Sumit, good evening. Okay, now, so first we'll go in a proper manner. Okay, we'll go in a very proper manner where we'll discuss everything. We'll discuss about cell cycle, we'll discuss about different parts of the cell cycle, and then we'll concentrate on the mitotic part. Okay, so what is the cell cycle? The cell cycle is the sequence of cell growth and division. Okay, it's a cycle, as I said, it's a cyclic process. So there's a sequence of events which happens. So that sequence is known as cell cycle. It's the period from the beginning of one division to the beginning of the next. Okay, it takes time to complete one cycle and it's called the generation time. The time in which the cell is completing one division is known as the generation time. So it has three phases. There is a G1 phase, there is an S phase, there is a G2 phase, and then there is M phase. G1 and G2 is together known as interphase. And M is the mitotic phase, that is a given. We know that it has 4 parts and everything. Okay, that we'll discuss. Now, G1 as G2 is when the cell is getting ready to get divided. Okay, it's the interphase. It's when the cell is getting ready, it's readying itself to get divided further. That is the interphase. Now, so as I said, beginning of one division to the next. So there is interphase, G1, S, G2, and M. M is the mitotic phase, which is again further divided. And there is the cytokinesis where the cytoplasm divides. This is the interphase. Okay. Now, please understand it that for each and every cell, the amount, the generation time, the amount of time is actually, it actually varies from cell to cell. Okay, now cell cycles can range in length from as short as 30 minutes. It can be 30 minutes in a cleaving frog embryo. Like in a frog embryo, it divides every 30 minutes. Okay, again, why? Because it does not have proper G1 and G2 phases and all. Again, it takes several months in slowly growing tissue such as a mammalian liver. A liver takes very long time to divide, such as several months. So it varies, the time varies. Okay, so it can be 30 minutes also, it can be few months also. It depends. It varies from cell to cell. Now, the other thing you have to note is cells that are no longer capable of division, whether temporary or permanently, they go to G0 phase. Okay, a cell must receive growth promoting signal to proceed from the G0 to G1. See, the thing is, okay, G1, S, G2, and there is the M phase. So this is G1, S, G2. Now sometimes from G1, sorry, this thing is gone. Okay, I hope you guys can hear me. The thing is, from G1, sometimes the cell goes into G0 phase. Okay, why G0 phase? Because it goes into the G0 phase, because it goes into an arrest. Okay, so this is like when the cell is no longer, they can go for a cell division, they go into an arrested state. Okay, they can come back again. Okay, it's not that they're going only, they can come back again, but mostly they stay there sometimes, and they can come back again to G2. Sorry, because I have some problems. Okay, so what happens in interphase? So what happens during interphase? So during interphase, what happens is these three things: G1, S, G2. So in this stage, as I said, the cell is readying itself. Okay, the cell is readying itself to go for the cell division. The nuclear envelope remains intact. Okay, so here, as you can see, the nuclear envelope, it remains intact. The chromosomes occur in form of diffused, long, coiled, and indistinctly visible chromatin fibers. Okay, and the DNA amount becomes double. Here, the DNA, the genetic material, becomes double. Due to the accumulation of ribosomal RNA and ribosomal proteins in the nucleus, the size of the latter is increased. The genetic material is basically, it is increasing. Okay, because the cell is readying itself, so the genetic material is increasing. And the other thing is, a daughter pair of centrioles originates. Okay, here, this thing, these are the ones which create the spindle fibers. So they appear. Okay, they are the ones which are appearing. Now, the first thing is, as I said, G1 phase. This is right after M phase. Okay, this is right after the cell has divided. It is right after that. Now, G1 is kind of a resting phase because the cell has just now it has divided. So, it is kind of a resting place. It is also called as the first gap. G basically means gap. Okay, as known, DNA synthesis takes place during this stage. Okay, it is also known as the first growth phase since it has involved an RNA and everything it is involved. So it is known as the first growth phase also. Okay, here what happens, the chromatin is fully extended and you can't distinguish the chromatins and the chromatids and the chromosomes and all. It is not distinct. Genetic material is not distinct. Okay, now S and G2 phase. This is also an interphase only. This is also a part of interphase only. Okay, so what happens during the S phase or the synthetic phase? S means synthesis. So what is happening during this time only? All the metabolic activity, the DNA replication, everything and all, the metabolic activity is happening during this phase only. Okay, new histones are required in massive amounts. DNA is synthesized. Okay, each chromosome now has 2 DNA molecules. Okay, so what happens here? See, there is one chromatid. Okay, now it divides and then again it has 2 chromatids now. Okay, and finally in the second gap, that is the interphase, this thing G2 phase, synthesis of RNA and proteins. So here it is basically DNA replication is happening. Okay, here what is happening? Translation and transcription. This and all is happening in this phase. Okay, so now we'll come to the mitotic phase or mitosis. This is where the cell basically, we have the proper amount of cytoplasm, we have the proper amount of genetic material. Now we have everything. Now our cell is ready to divide. When the cell is ready to divide, it goes into mitosis. So mitosis is a process of cell division in which each of the 2 identical daughter cells receives a diploid complements like the C. In the S phase, the daughter cells became like this. The cells became, the genetic material became like this. Now it will divide. Okay, and go into their own cell. That happens in mitosis. Now mitosis again is followed by cytokinesis, where the cytoplasm divides. Okay, and mitosis is basically divided into 4 stages: prophase, metaphase, anaphase, and telophase. So there are 4 stages: P, M, A, T. Okay, now prophase. The first phase is prophase. Okay, till now we have seen that there are chromatins. Okay, we have chromosomes. We have 4 chromosomes here, which was basically happening due to the S phase of the cell cycle. We also had the spindle fibers, the centrioles we have, which was there, okay, which was formed during the G1 phase. Now what is happening? So beginning of prophase is marked by the condensation of chromosomes to form visibly distinct thin threads within the nucleus. Okay, so now the chromosomes are visible. Now, as I said, in some structures only you can see chromosomes. So now you can see chromosomes. Okay. Each chromosome is already longitudinally double. This happened in the S phase. Okay, and two closely associated subunits called the chromatids held together by the centromere. This was there. Okay, now each pair of chromatids is the product of duplication of one chromosome in the S period of Interphase. What happens is they are now, the prophase is just readying the cell so that the chromatids are now, chromosomes are now visible. Okay, and by the end of it, the nucleoli disappear and the nuclear envelope, the membrane, whatever membrane is here, no, this and all starts disintegrating. Okay, now metaphase. Metaphase, if the nuclear membrane and everything has disintegrated, now metaphase is what? Metaphase is where the chromatids, okay, the chromosomes, they come into the equator. As I said, equatorial plate, they come into one straight line. So what happens? The mitotic spindle forms, see here, the fibers have formed now properly, they have formed, okay, and consists of fiber-like bundles, microtubules, etc. Okay, each chromosome attached to several spindle fibers in the region of centromere. Okay, so there is a this thing, spindle fiber, and the centromere is holding the this thing. Centromere is holding the spindle fiber, and they come into in the middle. Okay, after chromosomes are attached to the spindle fibers, they move towards the center. Okay, and that center is known as the metaphase plate. Okay, so that place is known as metaphase plate. Now, anaphase, what happens in anaphase is, it was in the plate, now the centromeres divide longitudinally and the two sister chromatids of each chromosome move towards the opposite poles of the spindle here. So they are moving towards the pole. Okay, so the movements results from progressive shortening of the, what happens is the spindle fiber is actually making them move towards the whatever movement is happening here happens due to the spindle fiber. Okay, which pulls the chromosomes in opposite directions. At the completion of anaphase, the chromosomes lie in two groups. They are in the poles now, and it is the anaphase which actually, in which the cytokinesis starts. Okay, and finally the telophase. Telophase is the nuclear, here the nuclear envelope has formed. Okay, the nuclei is formed and the spindle disappears here. The spindle will finally disappear. The envelope has now come again. Okay, it is now covering again. The chromosomes undergo reversal of condensation until and then again they are no longer visible. Till now it was visible, now they won't be visible anymore. And finally these cells, they start going to the interphase. But before that, the cytoplasm divides. Okay, the cytoplasm divides, that is known as the cytokinesis, and now from the one mother cell, okay, it becomes into two daughter cells. Okay, and the other thing which you have to remember is the formula 2 to the power n. N is the number of times cell divides. Okay, so if a cell divides 2 times, okay, it becomes 2 to the power 2, that is 4 cells are formed. Okay, so that is the formula for mitosis. Number of cells formed is 2 to the power n. N is the number of cycles. Okay, so this is how basically the cell division happens. This is how it looks. Okay, now what is the significance of mitosis? There are a number of significance. It helps in maintaining the same number of chromosomes. It is responsible for growth and development. It helps in repairing damages, okay, maintaining proper size. It allows multicellular organism to maintain tissue, skin cells and all. It's important for sexual reproduction indirectly, not directly, because sexual reproduction directly, that is meiosis. Okay, and it allows, basically it is helping in growth and development of organisms. Okay, so if you have any doubts, just put it in the chat box. Now we just see some question and we can leave for today. DNA replication occurs in S phase, G phase, G2 phase, M phase. So DNA replication happens in the synthesis phase. That is the S phase. Okay, so the right answer is S phase. Next, during metaphase, mitosis chromosomes undergo coiling, move towards poles, line up at the equator, break and dislocate. See, metaphase is what? It comes to the middle, okay, or comes to the equator. Okay, so they line up at the equator. Next, the number of mitotic divisions required to produce 128 cells. See, as I said, the formula is 2 to the power n. Okay, so if 2 to the power n is 128, what is n? It is 7. Okay, you have to square root it and you'll get 7. So the answer is 7. Okay, so that's all for today. Okay, I'm sorry, because of a power cut, I had to switch off my camera. Okay, that won't happen in the next class. Just a second. Okay, so I hope you can see me now. Okay, so I hope you can see me now. My power has just come back. I'm sorry, because we had some power issues over here, and that is why I had to switch off my camera. I hope you learned something today, and in the next class, we'll be discussing about meiosis. Thank you, everyone. Bye-bye.